All right. Hello, everybody. Thank you all for having me here. My name is Bradley Voytek, and I am a professor at UC San Diego in the Department of Cognitive Science, the Haligio Data Science Institute, and part of the Neurosciences graduate program. I'm excited to be talking to you a little bit about the work that the folks in my lab are doing. Our lab sits at this interface between cognitive science, neuroscience, and data science. So here's all the current folks in the lab right now, and you can see we're a blend of different approaches. This is actually us in our new data science building here at UC San Diego. This is the new mural that just got set up, and I like this photo a lot. And before I begin going into the science, I want to thank you to all the support we've gotten over the years for the research I'm about to talk about from the Sloan Foundation, Whitehall Foundation, Kavli, NIH, and the National Science Foundation. All right, so what are we talking about today? I love brains. I love studying them. I love everything about them, trying to figure out how they work. This is where I begin. I also love oscillations. So if you stick an electrode in just about anything's brain, you'll see these rhythmic fluctuations in voltage sometimes. And these are neural oscillations, these beautifully recurring patterns of brain activity. So what's the problem? If I love brains and I love oscillations, let's just continue studying these things and have a good career, hopefully. The problem is I don't actually know what an oscillation is anymore. So I've been studying this stuff for 20-something years now and running my lab here in San Diego for the last 10. And I've managed to talk myself into not even knowing what I mean when I say an oscillation. And the part of the problem began when I was studying what these different oscillations putative functions are. What are they supposed to be doing in the brain? And if you go to Google Scholar or PubMed or your favorite peer-reviewed paper search engine, and you type in your favorite oscillation frequency band, delta, theta, alpha, beta, gamma, and you type in your animal model or cognitive behavioral state or disease of interest, and you type those together, you will see that just about everything is correlated with everything. And there are tens of thousands of papers on oscillations. And so I'm left just kind of wondering, what is an oscillation? What do we mean by a neural oscillation? So stepping back, I want to start by saying I really care about definitions and how we measure things in neuroscience in particular. I've worked in data science, in industry, I've been a data scientist, and some of what I've learned is how you turn your data into numbers that you can do statistics on is very critical. How you transform your data into numbers that you perform statistics on, very critical. This is so important. And the reason I say this over and over again, I think I even say it one more time, show the slide again in this talk later, is because we can't make inferences about the world unless we do this right. And so in my lab, we write mostly Python code and develop mathematical algorithms to help others understand, and to understand ourselves, how the brain works, whatever that might mean. So I'm going to step back and say, I want to give an example here. When I was a PhD student, there was this pretty transformative paper for me. This is out of Leon Duell's lab at Hebrew University, Shlomi Yuval Greenberg, in 2008 in Neuron. In this, they were looking at event-related neural activity to different visual objects. And so to just kind of orient you, if you're not familiar looking at these kinds of plots, this is time on the x-axis, frequency on the y-axis. And the color here is just how big the oscillation, putative oscillation, I'm going to hedge that a little bit, at that frequency is. So the more red it is, the bigger the power, the more power there is in that frequency band. The more blue, the less power there is in that frequency band relative to some pre-stimulus interval. So there's a lot of excitement around this time at oscillations in the sort of 40 to 60, 80 hertz-ish range, these gamma oscillations, and the potential mechanistic role in these gamma oscillations in coordinating neural activity and giving rise to perception. And so what Yuval Greenberg and colleagues showed in this Neuron paper is that you get this nice, clear event-related gamma activity in the 40 hertz range, which replicates a lot of previous work, but for particular kinds of objects, not for everything. And in the paper, what they very clearly demonstrated, and which has now been shown over and over again, is that if you look at this activity, in the trials, here on the bottom row, for objects like chairs, faces, or watches, if you only looked at the trials where people didn't make very tiny eye movements, not quite microsaccades, but mini-saccades, you don't see this event-related gamma. And if you only look at the trials where they did make these very subtle eye movements, you see this very broad high-frequency gamma band activity. And what they showed is that even though you have a lot of power in the gamma frequency range, replicating many studies over and over and over again, that it's likely that this gamma band activity in these kinds of experiments are not neural activity, but they're in fact artifacts driven by very small movements of the eye muscles, which manifest as gamma activity because eye muscle movement activity, those muscles, the signal that they give off, because muscles are also electrically active, is in this high frequency range. And this is true all the way back in the visual cortices. And so this really got me thinking, how do we know what we know from our data? How do we know if this very well replicated, well-trodden finding of gamma activity in visual cortical regions in EEG, two different kinds of visual percepts, is maybe not even neural? And so in my lab, we sort of take this view that we can't really talk about oscillations without first, one, understanding where they come from, and two, how they're traditionally measured. I don't have time in this talk to talk about where oscillations come from, but we have a lot of collaborations with folks in animal research and cell molecular biology, including Alison Moultrie here at UC San Diego, who develops human-induced pluripotent stem cell-derived cortical organoids. So you take a cheek swab sample from somebody, you coax those skin cells into becoming pluripotent stem cells and then grow them forward as neural tissue to see how they spontaneously wire up and sort of create this laminated neocortex-like structures that eventually, we've shown in previous work with Moultrie's lab, spontaneously generate neural oscillations. I don't have time today to go into that. I really want to focus instead today on how do we really measure oscillations. So this is a paper from former grad student Scott Cole in my research group, and sort of the charge was later led by former postdoc Natalie Shvarnikov and now Andrew Bender in my lab, looking at non-sinusoidal waveforms. So one of the first approaches we started taking in my group was saying, okay, well, you look at data and you can see that oscillations exist. Alpha, for example, visual cortical alpha is so striking and clear of an oscillation. You can see it in the raw data. Same with rodent hippocampal theta, which tends to actually overlap in mice and rats in this sort of 10 hertz range as alpha, but it's called theta. It's in the hippocampus. It's this nice, beautiful, very strong, clear rhythmic activity. And what Scott had shown in this was that these oscillatory waveforms, here in this case, this isn't visual alpha, this is beta 20-ish hertz oscillation in the motor cortex, invasively recorded from people undergoing surgery for deep brain stimulation implantation in Parkinson's disease. So for people who aren't familiar, Parkinson's disease is one of the very few neurological disorders where we know exactly what in the brain has changed. You've got a group of dopaminergic neurons in the substantia nigra that are dying off for whatever reason. And that loss of dopaminergic input into the sort of basoganglia frontal circuit leads to the Parkinsonian symptoms of resting tremor, bradykinesia, and so on. And also leads to an increase in beta power in the motor cortex. And our collaborator and colleague had previously published a number of papers on this, Phil Starr's group, Cora DeHemptine, showing that this 20 to 30 hertz beta oscillation is very pronounced in the motor cortex in these patients with Parkinson's disease, and that this lower frequency beta oscillation is phase amplitude coupled with high frequency gamma activity. What that means is you've got the phase of the low frequency beta, so if you're at the peak or the trough, say, that phase is co-modulated with the power in a high gamma frequency range. And specifically, during the rising phase of the beta, you see an increase in high frequency gamma power. And this high gamma activity, 80 to 150 hertz and above, has been repeatedly linked to population neuronal firing. So people have been excited in these intracranial studies because now you're under the skull, under the dura, right on top of the cortex, even sometimes penetrating the cortex in humans. And so you have minimal non-neural artifacts from things like eye movements that we showed previously. So you get really clear, beautiful event-related high gamma activity in a variety of tasks from these intracranial recordings. And that high gamma activity is coupled to the phase of these ongoing oscillations. And what Scott began to notice was that when he was looking at the raw beta activity, the raw neural time series data, here in white is one example I'm tracing here. I can see 1, 2, 3, 4, 5, 6 beta peaks within the span of about 350 milliseconds. And what he noticed is if you look at the full power spectrum, time resolved relative to the raw data, that when you have these sharper temporal deflections in the time domain, you also have these rhythmic bouts of very broad, high frequency activity. And so what Scott began to say was, well, hold on. Maybe you don't have two different oscillations, this high gamma power and this beta power that are coupled or co-modulated in this phase amplitude coupling manner. Maybe what's happening is that the gamma power is coming from the sharp temporal rise of the beta oscillation. So the Fourier transform, which is the method by which we, and it's related variants, right? So like wavelet analyses and things like this. The way that the math works out is we are asking the math of the Fourier transform and wavelet analysis to represent our time domain signals as a sum of sinusoidal features. And in order for the math to capture these very sharp, fast voltage changes in the time domain, in order for that to happen, you need increasing power in increasing frequency bands. And so these high temporal deflections, these sharp temporal deflections, are captured as high power in high frequency bands. It's like a very fast oscillation. And so an alternative hypothesis to this dual oscillation, beta is coupled to gamma, is that this apparent beta to gamma coupling is actually just a single non-sinusoidal beta process. And so it's one thing to make this argument from a theoretical standpoint or from a mathematical standpoint, but as I said earlier, we try and create mathematical tools, algorithms, and code to help other research groups better analyze their data and try and uncover whether something like these non-sinusoidal features matter. And so to do that, Scott and Tom Donahue and a number of folks in my lab, we created this bicycle Python package, which stands for the cycle by cycle analysis of neural oscillations. And to dive a little bit more into the details for folks who might be familiar with Python and things, what this does is it identifies in the time domain each individual oscillation waveform cycle. So here you can see a cycle is broken up by its trough to trough range, and we can then, for every single cycle, we create a pandas data frame where each row is a single oscillation cycle from one channel, from one person or animal, and each column is a different feature that we can derive from the time domain signal. So we can say, okay, when exactly did the trough occur? What was the amplitude of the signal, which is the average of the trough to peak and peak to trough? What's the period, which is just from one trough to the next? What is the rise time? How long did it take to go from the trough to the peak and then the peak back to the trough? What's the rise decay symmetry or the peak trough symmetry? So we over parameterize each individual oscillation waveform by a set of, I think it's a 20-something features. And so now for every channel, for every person, you have this rich data frame where every row is a single oscillatory cycle and every column is a feature for all of those cycles. And once we have this over parameterization, now armed with these tools, we can start to say, do any of these features map onto underlying physiology or disease? Okay, so I mentioned briefly the Fourier transform. I'm going to work within the Fourier domain here because that is the most common, but this applies also to wavelets and multi-tapers. And I'm going to say, okay, how are oscillations traditionally measured? So you have some kind of time series. It could be neural, it could be non-neural, doesn't matter. This works for all time series analyses. And we could say, okay, well, given this data, where is there an oscillation? And one way would be just to eyeball it. We can mark manually where we think an oscillation is. It's not good for scientific replication, so we will usually want to come up with some algorithms. But if I, as an expert, look at this and I think that this comes from human neocortex, I can say, well, maybe this is an oscillation burst of, I don't know, like 8 cycles or something like that. Maybe this is a longer burst of similar frequency band. Maybe this is a shorter burst of a higher frequency oscillation. I don't really know. And so that's insufficient. So what we usually want to do is we want to try and figure out mathematical tools and algorithms for reliably identifying things like an oscillation. So the historically traditional way is to use the Fourier transform. So to give you an intuition, by the way, my lab likes to create GIFs, animations for things. So this is from former PhD student Tom Donahue, just illustrating how the Fourier transform works. So here you've got a time domain signal in black, and we are going to then fit a series of sinusoids to that time domain signal, illustrated above here. But we can represent those sinusoids by 3 features. The starting phase of the sinusoid, which I'll be plotting here on the circle, as well as the power at each frequency. So here we go. You just start adding more and more sinusoids, and you start to get a better and better fit until you have a perfect mathematical decomposition of your time domain signal as a series of sinusoids. This is a lossless approach for parameterizing your data. And so what this tends to look like for neural data is here on the right. This is just another illustration that I really like, showing the same kind of thing, where you could have a complex appearing time domain signal. But if you actually do the sort of Fourier transform of it here, you move a voltage, for example, on the y-axis by time to a power by frequency signal, where this signal here in red in the bottom left is mostly composed of 3 sinusoids. Neural data tends to look more like this, where now what I'm doing is actually plotting this on log-log scale. So this is log frequency on the x-axis, so 10 to the 0, 1 hertz, 10 to the 1, 10 hertz, 10 to the 2, 100 hertz, by log power on the y-axis. And what you tend to get is this sort of shape here, where you have exponentially decaying power after about 10, 20 hertz or so. That appears as mostly linear in the log-log space, relatively flat power out to whatever that sort of frequency at which it bends down is. And then you have these little regions here where you have order of magnitude or more power in a narrow frequency band around and above whatever that background process is. And it's these things, these bumps in the power spectrum, that we normally traditionally define as oscillations. So Biyuha talks about this, Yuri Bujaki talks about this. In a power spectrum, brain oscillations appear as bumps on top of what Biyu and others have referred to this as 1/f slope. This sort of, I'm tracing here that sort of background process is called the 1/f component, because it is, power is 1/f, is proportional to 1/f, 1 over frequency. And so you have this 1/f component. And anything above and beyond that is an oscillation, these bumps. So, okay, what's the problem with this approach? Here we've got 2 bumps in a power spectrum. So log power by log frequency, 2 bumps, very clear. But we've got the 1/f component. And so my lab began to get stuck on what is that 1/f component. So the reason that we really cared about this, and we ended up developing the spectral parameterization tool, formerly known as FOOF, for fitting oscillations in 1/f, which we're moving away from, which I'll explain in a little bit. But the reason that we came up with this is, okay, in this image on the upper right, this is a power spectrum from average power spectrum, let's say, from a group of participants. In the dark gray here is the power spectrum after I showed the participants a visual stimulus, let's say. So let's pretend that this band here in blue is my 8 to 12 hertz alpha frequency band. I study visual working memory and attention, so I know that because I study visual working memory and attention, should also study visual cortical alpha, because that's a dominant oscillatory rhythm in visual cortex in humans. And so I run a task where I present people with a stimulus, and their activity goes from this sort of big bump in the alpha range here in light gray to a smaller bump in the dark gray. So I know that when I present a visual stimulus to someone, alpha power will decrease. Classic fighting, I know this. Whether or not you're aware of it, this is probably the model that you have in your head of what's happening to your data. If you do a Fourier transform of your data, averaged across all the people, you'll have a really big alpha bump beforehand. You present a stimulus and the alpha power is less. And that is a perfectly valid model. But here in the bottom left in number 2, we also know, for example, that alpha can speed up or slow down very strikingly across groups of participants, but even within people, as like Jason Samaha and others have shown. And so if I say that my alpha is 8 to 12 hertz, and I look at my 8 to 12 hertz window, but let's say that I present a stimulus to somebody and their alpha power doesn't drop, their alpha power just slows down. It shifts outside of my frequency window. The total alpha power like changes a lot in terms of like this window here, because only like 50% of my alpha is even in my a priori frequency window. But the change in the relative alpha power is nil. That relative to that background aperiodic 1/f process, the alpha power hasn't changed at all. Or alternatively, we also know that the entire power spectrum can shift up or down. What Kai Miller and other folks have adopted as these broadband spectral shifts. Again, if I'm only looking at this 8 to 12 hertz alpha window, it'll appear as though my alpha power has dropped, but that's not really the best way of describing. Technically, yes, I have less power in the alpha range, but I also have less power at every frequency in my signal. Or in condition number 4 here on the bottom right, we also know that the power spectrum can change its slope. Ella Poldvani with Rafael Malik has showed this, and others, that the that the entire power spectrum can shift. And again, if I'm only looking at this 8 to 12 hertz alpha window, I'll say that my alpha power has dropped. But again, maybe not the best way of describing the data. In other words, all 4 of these scenarios, which are totally physiologically plausible and can and do happen, they will give the exact same result using standard analysis methods if I'm only looking at like filtering in my alpha range and looking at the Hilbert transform to look at the change in analytic alpha amplitude, for example. Very common approach. That is, we can't adjudicate between any of these without more careful parameterization. So our solution, as with many things in my lab, is curve fitting using Python. And so we have this module that we created formerly, like I said, called FOOF. We're moving the name to SpecParam. One of the reasons why we're changing this is that this paper has been picked up by not only neuroscience, but actually quite a number of other fields. So it's been used now in looking at fusion reactor data from people working on fusion power, and groups of astronomers and cosmologists looking at spectral data from celestial objects. So those groups don't talk about oscillations in 1/f. Those are sort of neuroscience more specific terms. So we're creating this more generalizable term called SpecParam. And so the new SpecParam package has a lot of options now that the initial FOOF package did not have. And this initial version of FOOF SpecParam was spearheaded by Tom Donahue and very close friends and colleagues of mine, Matar Haller and Avgusta Sjestuk. Actually just hung out with Avgusta yesterday. Former postdoc in my lab, Eric Peterson, and stellar undergrads, former undergrads from the lab, Leonardo Madanda and Julio Dominguez. And so the way that this spectral parameterization works is it first does its best to try and fit this background, heavy quotes here, aperiodic is the term that we use, 1/f signal, and then it just subtracts it out. So you have the residuals from that initial fit. And the idea being that anything above and beyond that, the bumps above and beyond that background signal, are our putative oscillations. So we first check to make sure that the first identified highest peak in that is distinguishable from noise. So if it is greater than some noise threshold, plus or minus 2 standard deviations or what have you, we're going to call that a peak in the power spectrum, and we're going to fit a Gaussian. Now, the new SpecParam module allows you to actually swap in different functions. We use Gaussians because that was pretty common in the field to use Gaussian fitting. We then subtract that Gaussian fit, and from those residuals, find the next peak, ask if that peak is above our threshold. If so, fit another Gaussian, subtract that out. So you're iterating through. If the next peak is below the threshold, you stop fitting. You go back to that signal here in B, you fit both of those Gaussians simultaneously, subtract that out from the original power spectrum in A to get a cleaner estimate of the aperiodic signal. refit that, combine them all here in red. So with just a couple of parameters describing these two Gaussians, for example, and that background aperiodic process, we have a very good parameterization of our noisy power spectral signal. And so from this, we have a couple of features. One, we get that 1 over f exponent or slope. How fast does it fall off? The frequency at which it bends down, the knee frequency, the total offset of the signal, as well as the Gaussians, which are described by their center frequency, the power relative to the background, and their bandwidth. Okay, so we've got this set of parameterization. We can identify putative oscillations. We've got this background, aperiodic stuff. What do we mean by aperiodic activity? So I want to emphasize here that just because there's power in a frequency band does not imply that there is an oscillation at that frequency. This is known as the Fourier fallacy. This is going back all the way to, in human neuroscience, I'm sure it goes earlier, but in science in 1948, neurologist, maybe neurosurgeon Jasper says, even though it may be possible to analyze the complex forms of brainwaves in a number of different sine wave frequencies, this may lead only to what might be termed a Fourier fallacy, if one assumes ad hoc that all of the necessary frequencies actually occur as periodic phenomena in cell groups within the brain. That is, just because there is power in a frequency, you cannot then inverse that and assume that that means that there is an oscillator in the brain at that frequency. Why? Well, to give a very contrived example, and this is a paper from Tom Donahue, Natalie Shvarnikov, and myself back in 2021, giving recommendations for studying oscillations. If you look at a time series that is pure white noise, just Gaussian distributed white noise, where by definition, every point is independent from the next, it has equal power at all frequencies. So you would be very hard pressed to say that this signal here has an oscillation at 4 hertz. It doesn't. It's designed that way. Mathematically, it does not. But in order for the Fourier transform to capture this white noise time series, it has equal power, roughly equal power at all frequencies. Or at the other extreme, you can have something like a Dirac delta function, or in the discrete space, a Kronecker delta function, where you have a time series that is all zeros with one non-zero element. That has equal power also at all frequencies. And you'd be very hard pressed to say that there is an oscillation at 5 hertz in this kind of signal. And so this illustrates the fact that you can have non-oscillatory data, but by definition, because the math of the Fourier transform is doing what you're asking it to, it will always give you power at some frequencies in your signal. So here is an aperiodic only signal that we've constructed that is based off of different physiological features that we can simulate. And it will have this kind of 1 over f-like distribution of power, but again, by definition, no oscillations anywhere. But if you filter in the canonical frequency bands, it'll certainly give you what looks like even bursts of oscillations at different frequencies. So by construction, none of these signals have an oscillation, but of course, filtering them will return what appears to be a rhythmic signal. Okay, so what is aperiodic activity? This is a great paper from Kai Miller back in 2009. Walter Freeman had a paper with Jai back in 2009 also, showing that if we assume that our signals that we're picking up in EEG and LFP and so on are mostly driven by postsynaptic transmembrane currents, these currents have this double exponential form in the time domain. And if you do a Fourier transform of a double exponential in the time domain, you get this sort of 1 over f-like phenomena. Just that's the natural consequence of the math. And so what Kai Miller showed is that if you just assume that data recording are all of these presynaptic spikes are causing these postsynaptic excitatory currents that are all integrating into what we call EEG and LFP and so on. As you increase the total number of asynchronous spikes, your entire power spectrum will shift up or down. That is, the shape of the transmembrane currents driving the local field potential in EEG directly influences the shape of the power spectrum. To illustrate this in GIFified form, if we have a bunch of presynaptic spikes that we convolve with a synaptic kernel, here in pink, integrate all those together into our LFP and do a power spectrum. This is what it looks like here. And if we change the shape of the synaptic kernel, leaving all the spike times the exact same, what you'll see is that as you change the kernel to make it shorter and shorter in time, you get more and more high frequency activity. That is, the whole power spectrum begins to sort of bend and shift up. The knee gets pushed out. This is just a natural consequence of the math. And so what you're seeing here is that as you change the time constant of the synaptic kernels, you are pushing the knee farther and farther out in the frequency domain. So that leaves the question of, can we infer the postsynaptic current drive from the shape of the power spectrum. So former grad student in my lab, Richard Gao, published this paper in 2017 in our lab showing that if you do the same kind of model that Kai Miller had done, but now you have these asynchronous AMPA excitatory currents, as well as these inhibitory GABAergic-like currents. You see here that these are the currents in the time domain. They integrate with this asynchronous spiking to here in C. And if you do the power spectrum of these, you'll see that if a signal is purely driven by excitatory currents here, these AMPA-ergic currents, you can see that the knee here is much farther out in the frequency domain, whereas here in the purely GABA case, it is back here. And so the shape of the power spectrum at the two extremes between AMPA and GABA, you can maybe say that if we change the relative ratios of these AMPA and GABA that are giving rise to our signals, they will jointly integrate to this power spectrum here. So if you could look at the shape of the power spectrum, can you invert that? And in simulation and in some open existing data sets, Richard showed that this is possible. This does not mean that the shape of the power spectrum is exactly mapping to the relative contributions of excitation and inhibition, only that the contributions of excitation and inhibition do influence the shape of the power spectrum. There are many other kinds of transmembrane currents, H currents, I currents, neuromodulatory, and so on. And so it's not a one-to-one mapping. I just want to be very clear about that. But what we are trying to emphasize in our lab is that there may very well be so much more that our data want to tell us. We just need to know how to measure it. So from individual oscillatory cycle waveform features to all of these spectral features that we can parameterize. And with that, I want to say thank you, everybody. I wish I could be there in person, but unfortunately I cannot. And I hope it's a wonderful conference. And I will be at the Society for Neuroscience conference next month in Chicago. So if you want, please come and see us. My lab has a bunch of posters on Monday afternoon in one block together. So come say hi. I'll be there for 4 hours at least. All right, bye.